Alright, so I just went to the kitchen and got a couple of things. I got coffee beans, lentils, fennel seeds, sesame seeds, and dehydrated yeast. And you can see still everything is visible, pretty small, but definitely visible. But let's talk about the yeast. These are not individual yeast cells. Let's check it out. Alright, let's see how small can really be. And what I just showed you is that lots of things we can still see: coffee beans, sesame seeds, grain of rice. But then as we zoom in and get into the smaller world, our eyes fail us and we need to use microscopes. Grain of salt we can still see, but when it comes to an amoeba or other little organisms in a drop of water, we can't see that anymore. For example, an amoeba is about 0.5 millimeter size, big, and we can't see that anymore. With a light microscope or even with a dissecting microscope, we can definitely see amoeba, we can see paramecium, we can see other multicellular organisms like tardigrades and rotifers, and then we go even smaller. So when we look at our cheek smear, our skin cell from our mouth, we can definitely see cells, but we definitely need to go to higher magnifications like 100 or 400x magnification to actually see something, see the nucleus, see the structures and so on. And then it gets even smaller. The Baker's yeast in the image you saw, the Baker's yeast, what you can see, was dehydrated, but it's really not one cell. It's a bunch of cells clumped together. So actually one single cell of yeast is much smaller. It's just a couple of microns large. Alright, let's go even smaller because we are looking at bacteria and viruses. So once we come to our beloved E. coli bacterium, it's around a micron long and wide. It's like a slender rod, and our bacterial filters are 0.2 or 0.4 microns, so you can see that the filters can actually filter out E. coli. But as it comes to smaller things, it gets even more tricky. And this is actually a really good example because E. coli is a bacterium, and you can see that our mitochondria and our cells are pretty much the same size of a bacterium. And this is one of the aspects that confirms that mitochondria and chloroplast in plants are endosymbionts, so they derived from bacteria and were engulfed into an organism that then uses mitochondria and chloroplasts to produce energy. Anyway, let's go even smaller here. You can see now we are at the viral level, and the even smaller organisms, again, you just need an electron microscope at this point to see any structures at all. You get lots of different types of viruses, different sizes, different shapes. Phages are really interesting right now because they're using phages that are very specific to different bacteria for phage therapy to kill antimicrobial resistant bacteria. So that's going to be an interesting field. And again, let's go even smaller. Beyond the viruses, you see individual organelles, organelle sizes. Ribosomes, for example, are the protein. They're not organelles, they're just machines of producing proteins. So ribosomes occur in prokaryotes and eukaryotes, slightly different structures. And as we zoom in even more, we are getting to our RNAs, different parts of a cell. Antibodies obviously really important in immune responses. And then beyond these larger molecules, we get into different individual molecules. So we've got adenine, we've got glucose, sugar molecules, methionine, and then we get into the atoms. And now we are really down to small sizes. So a water molecule is 275 picomolar. It's tiny, tiny, tiny. And we can't really see these anymore with any scale. So again, let's zoom back out and let's understand what lies in front of us, even if we can't see it. Alright, how small is small? I would say really small.